I want to move forward our conversation to manufacturing and scalability. Um, and Julie, perhaps we could start with you. What would you say are the biggest technical or operational challenges that are faced in scaling iPSC-derived products under GMP conditions? And what would you say is the best way to address them, or what solutions perhaps are being explored?
Yeah, so thank you. Thanks for the question. I think scalability is one of the, very high level of, especially even differentiation protocols, because we have automation in place already, but we don't have necessarily an end-to-end system that allows us to do everything. So we have to figure out piece by piece what we can put together, what can work together. And as we're doing our cell line development and characterization, I think that there's a lot of challenges within that. And, we want to make sure that we have robust characterization, but being able to, take the samples out at certain times. I think one of the other important things is really now that we're seeing machine learning and AI and being able to help optimize that is have a system where we can draw off samples at timed intervals so that we can really help to optimize our process. So I would say, being able to scale it in an automated device that would be closed down. Um, Quality control parameters, I think that that's getting better. I don't know if, with iPSCs, but some of the protocols are getting shorter and shorter, and we want to be able to make sure that those release tests can accommodate the shorter, possibly manufacturing. Cost management, I did discuss. I think that that's really a piece, we have to get our arms around. So whether that's being able to manage these products from vein to vein in one type of system. We see some of that out there, like the Solaris model, where you put it in and it's in a cassette and then it comes out in a cassette and you just have to do a fill finish. I mean, are there opportunities for iPSCs with that? So I think these are some of the challenges that we're seeing. Cost, supply chain, I would say as well, but making sure that you always have a second vendor in place in case there is problems with supply.
Sure, sure. That's a really good point. Erin, I want to be able to, of course, offer you the same question. The biggest technical or operational challenges that you feel are the scaling of iPSC-derived products are facing under GMP conditions. What are you seeing?
Yeah, I mean, very similar pain points to what Julie mentioned. I think, the sort of the way I think about it is, these products are living cells, right? And so they will respond to different environmental cues, including how many times incubators open, how an operator handles or manipulates the cells. That can be, that can make a huge impact on the critical quality attributes. And so I think to echo what Julie said about, making sure that we try and standardize our manufacturing processes as much as possible, knowing those critical quality attributes and how slight perturbations may impact those CQAs is really important. And then, we don't have, or the field does not have at this point, an end-to-end, fully automated process, right? We can apply automation at different points in time, but particularly with differentiation protocols, we may need more bespoke automation at specific points in time. I think that there's a lot of exciting advancements in that right now, and I think we're slowly getting there as an industry, but there's common pain points that I think all of us can relate to in terms of scaling and standardization.
Sure, sure. Erin, how are you approaching, or in general, via Astellas, what have you, the development of master cell banks to ensure consistency, quality, and global regulatory compliance? And I should note that our poll results for the second poll question just came in, and the biggest barrier to scaling iPSC therapies at almost pushing 40% is in fact the regulatory complexity and uncertainty. So probably a good time for this question.
Yeah, I mean, we've been in this space for a while, and so I think we're able to leverage our past experience and the engagement with regulatory agencies on the paths that we're looking for. I think also the application of next-gen sequencing to help with adventitious agent testing and the ability to really characterize those master cell banks is critically important. We also have invested in and established state-of-the-art GMP manufacturing facility here in Massachusetts, and so I think it's it's something that we're taking taking a careful path in order to inform how best to produce these in the future. But it's certainly, it's in many ways over the last several years it's been trailblazing, and so we've learned as we've gone along, and I think that's going to help as the field moves forward, learning what works and what doesn't, and then just paying more attention to things like genomic integrity and the different ways in which we can interrogate the quality of our master cell banks.
Sure. Julie, I'd like to offer the same opportunity to you to talk to us about the development of master cell banks. How do you feel it's being approached?
Yeah, no, a lot of things I would echo with what Erin is saying. Focus on, first of all, selection of the cell source. I think she mentioned genetic stability, and then the functional assays that we're using. Obviously, we're in a clean room. We have about 16 clean rooms across our 3 sites where we would be scaling that up. We actually do it in a pilot facility before it goes into the clean room, so we have opportunities to do engineering runs within that environment as well. But maintaining, I would say, to control consistency and to control the variability of these, using validated reagents. One good point that Erin brought up about the FDA and regulatory, that is critical, and the early steps. I think that's one of the things sometimes that investigators might say, oh no, we can just take it to an IND or we can take it to a pre-IND. There's opportunities before that, and interact becomes quite important. There's also other steps where they might accept a CAT, a CATT, and that allows you to maybe put in 2 pages of a platform technology and get back a few questions on that within a short period of time. So there's certainly ways to be able to get more information. I think that's important as you're in this innovative space, which is definitely not locked in. And as we're discussing, there's more automation that's needed. There's more information that we can gather possibly through AI and collecting more data. So it really does help for the regulators to be able to weigh in. They're the ones that are going to approve this for us to go forward. So, I mean, as much as possible, continuous monitoring these with the appropriate tests to be able to improve your outcome of the final product.